everyone, and welcome to today's live broadcast. IQCP is a four-letter word, presented by Dr. Stephen Cotten. We're excited to bring you this educational web seminar presented by Labroots, the leading scientific social networking website and provider of virtual events and webinars, advancing scientific collaboration and learning. For more information, please go to www.labroots.com. I'm Christina Jewell of Labroots, and I'll be your moderator for today's event. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click the green Q&A button located at the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located at the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know you're having a problem. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your webpage and follow the process of obtaining your credits. Now I'd like to introduce Dr. Stephen Cotten. Dr. Cotten is Director for Chemistry, Immunology, Toxicology, and Point-of-Care Testing at the Ohio State University Wexner Medical Center and Assistant Professor in Pathology at the Ohio State University. He received his PhD in Pharmaceutical Sciences from the School of Pharmacy at the University of North Carolina at Chapel Hill, where he also completed his training in clinical chemistry in the Department of Pathology. Dr. Cotten is board certified in clinical chemistry by the American Board of Clinical Chemistry. He has also served as the chair of the AACC Ohio Valley Local Section and is currently the newsletter editor and online community moderator for the new Mass Spectrometry and Separation Sciences, MS3, division of the American Association for Clinical Chemistry. His primary research interests include drug testing in alternative matrices and operational efficiency metrics for clinical laboratories. Please join me in welcoming Dr. Stephen Cotten. I will now turn the presentation over to him. Dr. Cotten?
Thank you for that wonderful introduction. So today we're going to talk about IQCP and its impact on laboratories. So what does IQCP mean to you? This has been a topic that has provided a lot of stress for laboratories, and there's been some creative acronyms associated with this. One of them is idiots questioning competent professionals, I question confusing policies, indiscriminate quagmire creating panic, I quit clinical pathology, or imposed quality control plan. But most of you probably know that it stands for individualized quality control plans. So just an outline of what we're going to cover. We're first going to begin conventional approaches to quality control in the lab, and then we're going to discuss CMS changes to quality control, what that means for the affected test, and then how to perform an IQCP, and then finally end with what does the College of American Pathology have to say about IQCP, what is in their checklist. So a little bit of background. Guidance for quality control for clinical laboratories has conventionally stated two things. It is said that 2 levels of external quality control must be performed every 24 hours, or you must follow the manufacturer's instructions for quality control. However, this can create some conflicts if the manufacturers do not recommend external controls at the same frequency of every 24 hours. And so this discrepancy has largely gone unenforced, as there are many tests and many instrument platforms which are moderately or highly complex that actually don't meet these CLIA requirements. And effective January 1st, 2016, if labs wished to remain compliant, the practice of running QC less frequently, they must perform an IQCP. And so the IQCP involves three things. It involves a risk assessment, a quality control plan, and a quality assessment. Now, labs have shared a lot of frustrations regarding about this because this concept of IQCP is relatively abstract and it's relatively new. And so a lot of labs have simply said, well, I wish I could understand this better. Just tell me what I need to do. Don't make me think about something. How can a small hospital with limited resources perform the appropriate studies needed to implement an IQCP? There has been very little training, and the instructions have been very confusing. The amount of work required is often very daunting, and the guidance is vague and abstract. So it's very frustrating for something so important with such poorly defined expectations. And probably most importantly, labs have said, we haven't had enough time to do this. So let's go into some of the things that CMS has changed about quality control. The initial notification that there was going to be a change to quality control starts in 2011. This is where the initial notification that EQC, or equivalent quality control, would be phased out and then replaced with a lab-specific risk-based QC approach. Flash forward 2 years, CMS releases a bulletin better describing these QC changes. They discuss IQCP training and the transition process that will be effective January 1st, 2016, and will be phased in during a 2-year period. At that point, there will be no more EQC, equivalent QC, or electronic QC that will be valid. So what does IQCP mean for you? It may be impractical to run 2 levels of external QC every day for your test. This can be due to just cost restraints, too many instruments, the methodology of the assay doesn't really limit itself to running 2 external levels. You might batch per cartridge or per kit or per specimen, or there could be just a lack of control material. And so IQCP gives labs the flexibility to meet these regulatory QC requirements that are appropriate for your test system, your patient population, and your test environment. It's important to note that IQCP is technically voluntary, but the implementation is needed if you run less than 2 levels of external control every day of patient testing. Some of these subspecialties that have been excluded from IQCP include anatomic pathology, histopathology, oral pathology, and cytology. There are also some interesting nuances with tests with multiple subspecialties. So under CLIA, certain tests may be assigned one of several subspecialties. If a test is eligible for an IQCP based on any of the subspecialties, then the laboratory may elect to use an IQCP approach for that test, regardless of the subspecialty they have assigned to it in their laboratory. For example, fish testing may be classified as either histopathology or cytogenetics, and so the lab may have it under histopathology on their CLIA certificate. If so, the lab can still use an IQCP for their fish testing. So what are some of the responsibilities of the CLIA lab director? First and foremost, the director is responsible for accurate and reliable test results that are appropriate for patient care, no matter what QC method or approach is used. They are also responsible for ensuring that the IQCP meets the requirements set forth in the IQCP interpretive guidelines. They are also responsible for signing and dating the IQCP. This cannot be delegated. So now that we've reviewed the CMS changes to QC, let's talk about some of the affected tests. what tests might be included in this IQCP program. A lot of these are actually point of care tests. Some tests that currently use electronic quality control methods like ISTATs, microbiology testing, blood gases, moderately complex kit testing, ACT or clotting, and anywhere where manufacturer QC is less stringent than CLIA requirements. So I've broken that down into a little more detail here. For blood gas instruments, internal or electronic QC is most often used, and these instruments can be deployed throughout the hospital. They can be a part of respiratory therapy, which has multiple sites throughout the hospital, but they can also be found in operating rooms and the ED. For microbiology testing, susceptibility testing on the microscan, gene expert testing for C. diff, and commercially prepared media may not actually have 2 levels of external control run daily. In addition, you can have rapid molecular tests for flu, serum specimens on point-of-care HCG kits, and quantiferon testing for TB. What tests does IQCP not include? This is not a comprehensive list. It is the responsibility of the laboratory to audit all of their tests to see if they currently meet the criteria of 2 levels of external QC every day of patient testing. And we'll talk more about this later. So we're going to go into the meat of the presentation. How do we actually perform an IQCP? Well, CMS has provided an IQCP workbook. It is available for download online at www.cms.gov, and it outlines the steps involved to develop an IQCP that meets the requirements set forth by CMS. The IQCP, or Individualized Quality Control Plan, has 3 major components. It is composed of a risk assessment that looks at what can go wrong with this particular test system. That is the starting point of the IQCP. Once you've performed the risk assessment and identified the risks involved with that testing, you can develop a quality control plan. So how do we prevent or detect errors from happening for the risks that we've identified? And then the third step is a quality assessment. Is the quality control plan that we've put in place effective for actually identifying and preventing errors in our test system? Our labs are probably already addressing some, if not most of these things in our QI and QM programs. So the IQCP is really just a formal process of collating all of your efforts together in a single cohesive plan. Let's begin with the risk assessment. So the first part of the risk assessment is to really look at the total testing process. There are 3 stages and 5 areas that are required for laboratories to evaluate. The 3 stages are the pre-analytical phase, the analytical phase, and the post-analytical phase. For the 5 areas, they can be divided into those 3 stages. These include looking at the specimen, the test system, the reagents, the environment, and the testing personnel. By looking at the 3 stages and the 5 areas together, you evaluate the total testing process to look at the risks associated with that test or test system. So some of the 5 required areas, if we look at some areas related to the specimen, are there specifics related to patient preparation? Are there issues with specimen collection and labeling? Are there issues with specimen storage and stability and transport? Are there specifics related to processing that can affect the outcome? For the test system, are there adequate systems in place to identify clotted specimens, to look at calibration issues, mechanical failure, optical failure, or built-in procedural or electronic controls that give you information about the performance of the test system? Additionally, what about the reagents? Do you need to look at shipping and receiving, storage conditions, preparation or expiration of the reagents. And then the environment. Are there temperature, humidity requirements, altitude or dust requirements that may impact the accuracy of your test system? And finally, labs should evaluate their testing personnel. Do they need specific training? Do they need regular competency? Are there unique educational requirements and experience needed? And staffing. Do you have a high turnover of staff in certain areas that may impact their training and competency over other areas? CMS has provided a worksheet to evaluate this risk assessment in these 5 areas. And so what's shown here are the pre-analytical, analytical, and post-analytical phases going across the top, followed by the 5 stages: specimen, test system, reagent, environment, and testing personnel. A good way to evaluate the consequence of your risk is a risk hazard matrix. This is actually not specific to IQCP, but it can be used to classify the degree of risk that you may have identified. And the 2 areas that you want to look at are the likelihood that an event will happen versus the potential consequence that an event will happen. So if the likelihood is high that you will have a very severe event, that would be considered a high-risk error. But if you have a very low likelihood that an event will occur, and if it does, the potential consequence is low, that would be categorized as a low-risk event. So let's walk through a blood gas example here. So let's say we've identified for the specimen, we can have issues of mislabeled specimens, and we can have issues of clots. Can we put a system in place that can detect and identify this? Yes. Well, we could implement barcode scannings, and we have a standard operating procedure for the collection that clearly identifies labeling of the specimen. For this test system, if calibration is expired, does that impact the results? Yes. Well, can we have a system in place that identifies that? Yes. The instrument has automatic lockout and automatic calibration. For the reagent, what is the impact of a degraded reagent to impact your results? Can we identify this? Yes. We can use QC, whether it's internal or external QC, to evaluate the stability of the reagent. And let's say your test system is impacted by excess humidity. Do we have an ability to detect excess humidity and prevent it? Yes. We can keep a daily log of the humidity levels in the lab and then recognize when the humidity requirements are not met for that test system. And for the testing personnel, if you have inexperienced technologists, would that impact the results? Yes. Well, can you do something to mitigate that risk? We can implement training and competency and even have them review the IQCP risk assessment that we've done for that test system. And then we can go back and classify the likelihood or the risk hazard for each individual risk that we've identified. And I've given some just examples here that maybe we think that the specimens are medium, the expired calibration is very low due to the lockout, the reagent is medium, the environment might be high, maybe we have a high issue, a big issue with humidity in our lab, and then inexperienced technologists would be low because we're confident in our training and competency. The historical data can actually be used to classify the risk and justify their classification. So it's important for labs to go back and review their own historical data. So using historical data, the I in IQCP, your own laboratory data is the support for your risk assessment and your QC plan. You can look at the number of instruments that have sent back for repairs over the last 2 years, the number of failed PT specimens with discrepant reports, the number of event reports or service occurrences that have included that particular instrument or platform, the number of personnel and the turnover of personnel each year, the number of rejected specimens and their reason for rejection over the last year, the number of corrected reports, or your own internal QC data. So now that we've identified the risk and we've classified them based on importance, we can now design a quality control plan that meets the risks, let's say the medium and high risks, and creates a system to mitigate or identify those particular errors. As I said before, once you complete the risk assessment, you need to determine which risks need to be monitored or controlled, or have they already been addressed by the manufacturer. The quality control plan designed must incorporate all the possible QC procedures, not just traditional QC. The QC plan must include the following items: the number of QC, the type of QC, the frequency, and the acceptable criteria. Your QC plan can incorporate many different types of QC. This can be electronic controls, equipment maintenance logs, internal controls, personnel training and competency assessment, calibration data, proficiency testing, or other data specific to the test platform. There is no minimum frequency. However, no QC is unacceptable, and QC cannot be less than the manufacturer's recommendation. And the QC frequency that you propose must be supported by your risk assessment. So let's go back to the risks that we've identified in our example. From our risk assessment, we thought we had an issue with mislabeled specimens and clots. The type of quality control we want to implement is verification of the specimen label and verification of the specimen quality. We want to do this with each specimen. And then how are we going to track this and what are the acceptable criteria? We're going to refer to our specimen collection policy, and we're going to track all of our rejected specimens. In the instance of a degraded reagent, the type of quality control that we're going to use is an internal quality control. And this is going to be 2 levels every 8 hours, according to the manufacturer. And the results must be within the manufacturer's range, and these results must be documented. So for our other risk of excess humidity that we've identified, we're going to perform temperature and humidity checks in the lab. And this is going to be performed daily. And our acceptable criteria, according to the manufacturer, is going to be 30 to 80%. So we've taken the risks, we've developed a quality control plan, and now we're going to use the quality assessment to take all of the QC measures that we've established, and we're going to create a system to review and detect and improve the quality control plan. Let's take a moment and contrast quality control versus quality assessment. If quality control means recording temperatures, quality assessment means the review of the temperature logs. If quality control means documenting your QC results, then the quality assessment means reviewing QC results monthly or even daily. If your quality control is documenting personnel training, then your quality assessment would be reviewing and auditing your employee training records. Furthermore, documenting maintenance would be reviewing maintenance, and documenting competency would be reviewing and auditing your personnel files. So some of the documents that you may want to consider, and this is from CMS regarding their IQCP workbook, may include QC data sheets, Delta check logs, proficiency testing results, complaints or event reports from physicians, specimen collection logs, specimen rejection logs, critical values or panic values, turnaround time reports, temperature logs, maintenance logs, training logs, and FDA alerts. And so the quality assessment takes all of your QC measures that you've established, and you want to create a system to systematically, on a regular basis, review and detect and improve the quality plan that you've put in place. So here's an example of a quality assessment. So the QA activity would be supervisor review and instrument printouts that are reviewed in QC logs. That's done on a monthly basis. And the assessment, was there variation? If there was, yes, then you propose a corrective action. So in this case, for the corrective action, they've listed remedial training of testing personnel and then reassess the testing personnel performance. Their second QA monitor would be a competency assessment. This is done on an annual basis after the first year. If there was no variation, then you don't need to rewrite or review the assessment. If the QA monitor is the laboratory director reviews and signs the QC logs, if this is done quarterly and there's no variation from the established policy, then you don't need to provide a corrective action. So what are some of the IQCP resources and templates that you can use? You can use the templates provided by CMS, which are available online. However, many manufacturers have actually created templates and platforms for assays that are affected by IQCP. This primarily evaluates the risk assessment portion of the IQCP, and labs will still need to individualize the plan based on their specific needs. I would encourage you to reach out to your vendor for your support. I've listed 2 examples of companies' blood gas that are affected by IQCP, both of which have created IQCP templates, which can be used by laboratories. Additionally, the American Society of Microbiology has created PowerPoint templates and PDF templates for IQCP plans for commercial MIC antimicrobial susceptibility testing, because this is a test system that is affected by IQCP. So now we're going to look at what CAP says in their new checklist regarding the CMS proposed changes to IQCP. So what does CAP say about IQCP? There are currently 2 important documents on CAP's website. There is a flow chart for determination of eligibility to use an IQCP, and there is also a new IQCP CAP checklist item and forms. The flow chart determines whether an IQCP can be used based on the testing waived or non-waived status, the pathology subspecialty, the manufacturer's recommendation for QC frequency, and some information regarding microbiology testing. Let's look individually now at the CAP checklist items. There are 5 checklist items that were reviewed and approved by CMS in August of 2015. The 5 checklist items specifically deal with IQCP, and there are 2 IQCP related forms. The first one is that laboratories must now have and maintain an IQCP list. So the laboratory has identified all the tests that they have implemented an IQCP plan and completed the CAP forms for laboratories using individualized quality control plans. This is very similar to the laboratory developed test list that all laboratories must now have and maintain as well. Checklist item, form 1 is the list of IQCPs. This is the list of individualized quality control plans. And so you can see that the CAP is asking for 3 specific things. They're asking for the laboratory section or department that the IQCP lives, the test instrument and device. This includes the name of the instrument, the manufacturer, and the model. And then the tests are specific analytes that are included in this IQCP. So for instance, for a blood gas analyzer, one instrument may actually have multiple analytes that are impacted by IQCP. And these can be found on CAP's website, on eLab Solution Suite, accreditation forms and instructions. The second thing is an IQCP summary form. And so this is very similar to the form provided by CMS, but CAP has customized it a little bit for the inspection process. And so you can see it has the instrument device, including the name and manufacturer and model, the test affected, the number of devices in use at that CLIA site, and the number of test sites if there are more than one site at a particular CLIA number. And then you may recognize the 5 areas that were required in the risk assessment. And so the bottom has you list all of the processes that you've identified as being risks during your risk assessment. And these deal with reagents, environment, specimen, test system, testing personnel, and they've included an other category as well. So checklist item number 2 actually details a little bit more about the risk assessment. So it first says that the IQCP for a test device and instrument includes a risk assessment to evaluate the potential sources of error to include all of the following. And I've highlighted 3 things that are actually additional items relative to what is listed in the CMS document. The first thing is the intended medical use of the test system and impact if inaccurate results are reported, or clinical risk. And so if you remember when we talked about classifying risks as low, medium, or high, this is where CAP wants you to actually evaluate the clinical risk. And this is not detailed in the CMS regulations for IQCP. The other thing that is unique to CAP is that data from the laboratory's own environment, instrument equipment, performance, and testing personnel must be used in the risk assessment. So they are really stressing that laboratories use their own data to go back and look at historical data to evaluate these risks. And then the final thing is that the manufacturer's instructions and recommendations are actually included in the risk assessment. So if the manufacturer lists a specific item that can greatly impact the analytical performance of a test system, you need to really look at that in your risk assessment and make sure that that is addressed in your quality control plan. So the third new CAP checklist item for IQCP is the quality control plan approval. So in addition to writing a quality control plan, the IQCP has to be approved by the laboratory director prior to implementation. And it's important to note that this is not, cannot be delegated to a designee. It has to be approved by the laboratory director. So checklist item number 4, the elements of the quality control plan. So many of these mimic what is outlined in the CMS guidelines for the quality control plan. So you must monitor aspects based on the risk assessment that detail the number and the type of QC and the frequency of the quality control that you're going to use, the criteria for its acceptable performance, how are you going to monitor the test and environment and the reagents, the specimen quality, instrument calibration, training and competency. But the one thing that is actually unique to the CAP checklist item is that they want a provision for dealing with multiple identical devices or variations used under one IQCP. So you can't just monitor a single blood gas analyzer if you have 11 in your respiratory therapy department. So you have to have a system that actually evaluates these multiple identical devices. The other item that is of note is that external material, control material must be analyzed at least every 31 days and with new lots and shipments of reagents, or more frequently if indicated in the manufacturer's instructions. So even if the manufacturer does not suggest that you need to run QC every 31 days, CAP has outlined that external control material must be analyzed every 31 days. And so it's important, again, to make sure that you've addressed multiple identical devices. So we'll talk about that a little bit more and the nuances there. So there's a couple of different scenarios. Let's say you have the same instrument at the same CLIA site, under the same CLIA site at multiple locations. You can have one IQCP per test system or CLIA site, but the quality control plan must monitor all instruments at your CLIA site. You can have the same instrument at multiple CLIA sites within a healthcare system. In this case, multiple IQCPs are needed for each CLIA site. Portions of the risk assessment may be shared, but each laboratory's individual data must be used, and each CLIA director must sign their respective IQCP plan. Let's say you have multiple analytes on a single instrument, one IQCP per test system. The risk assessment must address the test system and each individual analyte. And then let's say you have the same analyte on multiple instruments. In this case, you would need a separate IQCP for each test system or instrument that is a unique manufacturer or model. So the new checklist item number 5 relates to the quality assessment. The majority of the things outlined in the CAP checklist are actually mirrors of what CMS says. The one thing that I want to point out here is that CAP has added that reapproval of the quality control plan by the laboratory director or designee is done at least annually. So this is not included in the CMS guidelines for IQCP, but it is included in the CAP guidelines. So just to compare and contrast what CMS has mandated about IQCP versus what CAP has added. For the risk assessment, CMS and CLIA said you must look at the specimen, the test system, the reagent, environment, and personnel. CAP has added that you need to evaluate the clinical risk, you need to include your laboratory data, and you need to also make sure you've looked at the manufacturer's instructions. For the quality control plan, CMS and CLIA has said you must explicitly define the number, the type, the frequency, and acceptable criteria for your QC. And CAP has added that you need to make sure you have provisions for multiple devices on one IQCP plan. For the quality assessment, CMS has simply said that you need to review the QC data that you have set forth in your QC plan. And CAP has added that you need to reapprove your QC plan annually by the laboratory director or designee, and you need a list of all IQCP tests at a specific CLIA site. And so the things listed in the right-hand column, again, are just additional CAP components that are not included in the CMS regulations. So how do we put it all together? Just to recap, we begin with a risk assessment, and we want to look at our pre-analytical, analytical, and post-analytical phases. We do that by looking at 5 different areas, including the specimen type, the operator, the reagents, the environment, and the test system. You're going to identify some risks from that risk assessment, and you're going to draw from your historical data and analyte and location-specific risks for your CLIA or laboratory site. And then you're going to take the risk that you've identified, and you're going to find a way to control those particular risks. So let's say we've identified 15 risks. We might be able to control or identify those risks with 5 types of quality control. And those are going to be put together into a quality control plan. So and now moving from the quality control plan to the quality assessment, we have our 5 controls. And for the quality assessment for CAP, they want to make sure that you review the quality control and instrument equipment and maintenance and function data. They want you to evaluate pre-analytic, analytic, and post-analytic errors. And they also want to make sure that you evaluate complaints from clinicians and other healthcare providers related to the quality of testing. You want to make sure that this data is reviewed monthly, that you have some corrective action if you find deviation, and that the quality control plan is reapproved annually by the director. So what are the benefits of IQCP? Well, is there a financial incentive? You could make an argument that there's actually a large cost avoidance by doing an IQCP. If you were to predict your annual QC cost of running 2 levels of external control every day of patient testing versus the predicted annual cost of just running controls per month on your number of instruments, you could actually calculate a cost avoidance number. And that really is something you can take to your administration and say, by performing an IQCP, I've actually saved money or avoided cost for the laboratory. The second thing is it provides evidence-based quality assurance. By looking at your historical data, you're actually taking a step back and looking at what are the risks specific to your lab and your test system, and do you have a quality plan in place that effectively identifies those risks. So in conclusion, we've talked about how we take the risk assessment to identify what can go wrong, design a quality control plan as a way to prevent or detect errors, use that quality control plan to then do a quality assessment to see, is the quality control plan working? So IQCP, it gives labs flexibility to design their QC programs, and IQCP is voluntary. The goal is really to monitor the total testing process, and it should provide a continuous cycle of risk assessment, measurement, and review. And with that, I believe we will take some questions.
Thank you, Dr. Stephen Cotten, for that informative presentation. It's time for Q&A. If you have a question you'd like to ask Dr. Cotten, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click send. We'll answer as many questions as we have time for. Now let's get started. Dr. Cotten, our first question is, what documentation needs re-signed if an IQCP changes?
That's great. So I think according to, great question. So according to CAP, if you make a change to the IQCP plan itself, the plan does need to be re-signed, but you don't have to go back and re-sign the risk assessment.
Next question. Has anything changed since IQCP was implemented? Will AP ever be subject to IQCP?
That's actually a great question too. So just very recently, CAP has released an updated checklist, and I did see a couple of changes. The first one was related to commercial media for microbiology. I believe now is exempt from having to do an IQCP. The second one relates to fish, as I mentioned about subspecialty classification. And I believe that CAP has actually revised their stance on whether or not fish testing actually needs an IQCP. And so I would encourage you to look at your, if you are a CAP accredited laboratory, to look at your newest revised checklist. And I believe that both fish and microbiology media have been removed from, or are now exempt from having to do an IQCP.
Dr. Cotten, how do you address multiple instruments, multiple locations in an efficient IQCP plan?
Yeah, that's a great question as well. So at our institution, we certainly have labs that are spread out over multiple sites. We have a very large respiratory therapy department, and we also have testing that's performed at different CLIA sites. And so what we have found most efficient is actually having one site do the majority of the risk assessment and then share that with the other sites. And then the other sites will then take the risk assessment and then insert their own historical data as well as location-specific risks to really create an individualized risk assessment. And we really feel like that by having each site look at their own historical data, it is actually a really good way to develop a very smart quality control plan that really does a good job at catching errors. It is certainly a little more work, but you do avoid reinventing the wheel completely, because if one site kind of takes the lead to design the risk assessment, to sort of take a deep dive into that particular test system, and then the other sites can then go back and say, okay, well, I didn't really think about that, but let me add on my specific location risk or my specific issues related to testing at my site. It does actually save you some time, certainly. And we found that for IQCPs that we have very similar testing across the healthcare system, the majority of the time the quality control plans are actually very similar. So we are able to sort of take this a single design, individualize it some for the risk assessment, but then the quality control plan tends to be the same.
We have great questions coming in, but we're almost out of time. This will have to be our last. Dr. Cotten, what is required as part of the annual evaluation for IQCP?
Sure. So as I mentioned before, that the laboratory director does have to re-sign the quality control plan annually. It's also really important, if you do make a change, as the first question hinted at, that you do reevaluate your quality control plan. And obviously, if you do change test systems within that particular year, whether you're swapping out kits from a different vendor, then you would actually need to redo the risk assessment if you're actually changing vendor platform or a manufacturer.
I would like to once again thank Dr. Stephen Cotten for his presentation. Dr. Cotten, do you have any final comments for our audience?
I would just say that IQCP, once you've got one under your belt, it's really not that scary. As long as you sit down with everyone and maybe work through a template that can be adopted by your lab, IQCP should be a friend, not a foe.
Thank you once again, Dr. Cotten. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through February 2017. You will receive an email from Labroots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. We thank you for joining us and hope to see you again soon. Goodbye.